Welcome back, everybody. In this lecture, we'll be closing out our discussion of biological psychology topics when we cover a very critical topic to understand how the mind works called sensation and the eye. Now, it's probably not the last time you learn about biological mechanisms related to psychology. In fact, if you haven't gone over the touch section yet, you'll still get to learn a little bit more about other things that are going on in our body to help our mind work the way it does. But this is a really critical topic to cover, not only because of how applicable it is to our lives and how important it is to understand how the eye and mind interact to do what it is capable of doing, but it's also something that's really critical to cover because it serves as sort of a transition point into a new branch of psychology. What am I talking about when I discuss this transition? Well, what we're going to be looking at today, as I mentioned, is the process of something called sensation, the ability to convert some type of energy in the environment into some type of neural response that eventually travels from whatever area of the body is picking up on these energies to the brain so we can start to process it. And then we will be, after covering this concept of sensation in our next class, looking at something called perception, how the mind and brain is able to actually break down those signals coming from all the different parts of the body to the brain and make sense of those signals and give us all of the nuances that we are able to kind of break down when these sensations come from our body. We are going to spend an entire class on the topic of perception, and I promise we're going to break it down in a way that hopefully really gives you a robust understanding of both sides of these things. But to understand perception, we first have to go through the process of sensation and really break down what it is that our body is, well, breaking down and how it eventually gets to our brain in a specific way. So let's look at this topic of sensation in this class. As I mentioned earlier, we're going to focus our attention in this class on the eyes and how they're able to take a very special type of energy we're going to talk about in the next slide called electromagnetic energy and convert it into some type of neural signal that our brain can eventually break down. But before we talk about our eyes and focus our attention on that in this class, I do want to mention that it's certainly not the only what we call sensory organ that's out there that helps us process all of the stimuli in our environment. We also have really cool structures within our ears that allow us to take small changes in air pressure that are present in the environment and convert it into some type of neural signal that we eventually consider sound when we talk about how the brain processes it. And in fact, if you get into a biological psych class, you'll learn about these tiny hair follicles that are hidden in this structure that we call the cochlea that are actually able to send different patterns of responses to the brain that help us determine things like pitch and volume and timbre and all these other nuances to sound that we maybe take for granted, but neuroscientists have been able to break down on a cellular level. This is also true for our amazing system of touch that has the ability to take energies that exist in a lot of different forms, like tension, like temperature, changes in just pressure, and through our ability to kind of break down oftentimes chemicals or other things in the environment, and we're able to create some type of neural signal that eventually reaches the brain, and we can perceive not only what it is that we're feeling, but where we're feeling specific things. This is also true for our nose and tongue, which are amazing chemical breakdown machines that allow us to get all of these different complex molecules that are landing on our nose or tongue and kind of create this sense of taste or smell that many of us appreciate on a regular basis. If you take a biopsych class, you'll learn about most of these. You're also, like as I mentioned earlier, I'm going to learn a little bit more about touch in your section lecture a little bit later. But we have to sort of parse things out in this class so we can get to everything. And the way to do that for this is to focus on one particular sense. And as I mentioned, we're going to be focusing our attention on the sense of vision. Now, I know this might seem sort of arbitrary or random, but there's a lot of reasons why we tend to focus on vision in an intro class. One of the big reasons why we focus on it is we know a heck of a lot more about vision and how it works than we do with most of our senses. Another reason why we tend to focus on it is because there is a lot of kind of energy in our brain used to break down all the elements to vision that exist. So I understand that some of you might want to learn more about other senses, and I promise if you take other classes, you might potentially learn about those things. But for this class, just in terms of time and necessity, we're going to focus our attention on vision. And we're going to start with the sources of the electromagnetic energy that I mentioned earlier. For those of you that need a quick refresher, what we have in our environment are all of these different things called electromagnetic sources. Essentially things that produce energy, shoot out bursts of energy as a result of some type of energy conversion. And these electromagnetic surges are oftentimes measured in things that we call pulses. So from these sources of electromagnetic energy, the sun being the primary one, these pulses of electromagnetic energy are coming out, and they vary in both the wavelength, or the distance between the pulses, and the amplitude, and the strength of the pulse itself. And these different elements of that electromagnetic energy is what our eyes is trying to convert into something that will eventually make up as vision. What is it then that are the two properties that our eyes are most attuned to when we talk about vision? Well, as I mentioned, our eyes are looking for electromagnetic energy that vary in both wavelength and amplitude. In terms of what these things do, wavelength is oftentimes linked to, well, it is linked to color or hue of what it is we're seeing. The brightness of something is tied to what we call the amplitude of the light that's presented. Now, oftentimes, if you look at textbooks that describe how vision works, you'll see people draw wavelengths as these wavy lines like you see in the picture in the lower left-hand corner, measuring the wavelength of light as the amount of space between the tops or the peaks of the wavy lines. I'm not a big fan of those visual representations of wavelength because it sort of gives this misperception that electromagnetic energy is somehow snaking out of electromagnetic sources and constantly bombarding our eyes with this sort of snake-like structure that's entering in. In reality, wavelengths are about distances between pulses hitting our eyes. The reason why it's tough to represent wavelengths in this particular manner is that the distances between these wavelengths that help us determine color are microscopic. In fact, what we tend to do is measure wavelengths in something called nanometers, very small distances between the wavelengths that hit us. Short wavelengths tend to be linked to things that are very densely packed and come at very fast rates in terms of not how fast the light is moving, but the successions of the light waves that are hitting us. And long wavelengths, again, long is sort of relative for this, are the ones that are hitting us at a slightly slower rate. The amplitude of light, much like the wavelength of light, is often represented by some snaky line that varies now in this case in height. Again, not a great representation of what amplitude is about. It's more about the density of the light waves hitting us. Essentially, the more powerful electromagnetic source is, the denser the light waves are that are going to be hitting the backs of our eyes and eventually converted into some type of neural signal. Now, when we talk about frequency of wavelengths, what we see is that there's actually a large number of differences between different wavelengths that exist in the world around us. As I mentioned, we tend to measure wavelengths in a very microscopic distance that we call nanometers, just because those numbers are a little bit easier to work with than saying it's, 1/1,000,000th of a meter. If we do measure wavelengths in these nanometers, what we see is that the human eye has the ability to typically process light that's existing between 350 and 700 nanometers. Remember, that means the distance between the pulses is either 350 or to 700 nanometers. Just as a reminder, since these are distances between wavelengths, that means that if something is at, say, a 400 frequency in terms of light, that means that the light waves are actually hitting us much faster, or at a much faster rate, than if, say, something is at 700 nanometers. There's a little bit more distance, which means that the rate at which that light is hitting us is slow. Again, slow, fast is sort of arbitrary based on what we're looking at here, but that's how essentially our eyes are processing the visual light that exists in our environment. It's also important to note that there are varying light distances that our eyes aren't capable of picking up on. There's tons of electromagnetic pulses that exist in much higher and lower frequencies, and these things are utilized for a wide array of different things. It's just our eyes aren't capable of picking up on those things. And for those of you wondering, there are certain species that are more receptive to longer wavelengths and shorter wavelengths, and they do technically have the ability to see different types of light that we're not capable of seeing. But as human beings, we see a decent range, and it does allow us to see the visible light spectrum that we tend to see on rainbows and other types of visual representations that are around us. So even though it might be limited if you look at the grand scale of things, it's sufficient in terms of our ability to pick different colors and wavelengths of light that are around us. But how is it that we're able to actually convert this energy into some type of neural signal? Well, the cells responsible for this are actually found in the backs of our eyes, something that many people who study biology are sort of surprised by. They expect that we're processing light right in the front of our eyes, and that information is kind of creating this big signal and eventually goes straight to our brain. In reality, light takes quite the circuitous path through our eyes until it reaches the back, where we have these specialized cells that convert different nuances of this electromagnetic energy into different types of signals that are then sent to the back of the brain so we can start to process what it is our eyes were processing. We're going to get to that whole big processing thing a little bit later, and in fact we're going to get to those cells that are responsible for this conversion of electromagnetic energy into some type of neural signal a little bit later as well. Because what I'd like to do first is talk about the stops along the way that are critical in the kind of creation or the augmentation of the electromagnetic energy that is coming from all these different sources that are out there. And to understand this a little bit better, I want to start at really the place where everything sort of comes to a head, we could say. It's a structure in the eye that's not actually a physical structure. It's something that we call the pupil. Now, if we look into another person's eyes, the pupil is that little black dot that we have in the middle of all of our eyes. The reason why I say it's a structure that's not really a structure is because the pupil is actually just a hole. It's the hole that light passes through, so it has the ability to eventually reach the back of our eyes. And most of you have probably noticed along the way that the pupil, this black dot, changes in size in different environments. And this is a byproduct of the function of something that surrounds the pupil, a structure called the iris, which is sort of a muscle that has the ability to expand or shrink depending upon what's going on in our mind and the messages being sent to it. Now, the primary thing that the iris is linked to, if we want to understand kind of what its main function is the amount of ambient light or the amplitude of the light around us. If it's extremely bright outside, what the iris does is it expands, the muscle stretches out, and in doing so, it makes the pupil significantly smaller. This helps prevent sort of an overload from occurring in the back of our eyes where we're going to convert that electromagnetic energy into a neural signal. In essence, our eyes are pretty darn sensitive, and the cells that are back there require a really specific type of environments to be able to function optimally. And what the iris can do is sort of be the gatekeeper when there's too much information getting sent to the back of the eyes. So those cells that are very sensitive back there and continue functioning even when things are kind of overwhelming if too much was stimulated. If it gets dark out, what the iris has the ability to do is kind of compact itself, get thinner, and in doing so, make the pupil exceptionally large, and through this, allow more light to reach the back of the eyes. Now, I always love, if this was an in-person class, to do this really fun activity where we have students actually pair up with others and look into the eyes of some classmate as I turn the lights on and off. Now, it's kind of awkward, it's a little weird, but it does really highlight how responsive this iris is to the light around us. When the lights are off, everybody can usually see each other's eyes and see these giant pupils staring back at them. Then when we turn all the lights in the giant classroom on, the iris almost instantly expands and causes the pupil to get significantly smaller, thus preventing an overload of light hitting the back of the eyes. This kind of back and forth between the iris and the environment around us is a constant thing that we tend to take for granted, but it really does help us function. What was also really interesting to psychologists and biologists alike is the fact that we've now linked other things to the function of the iris as well. Even though ambient light is typically the thing that determines whether or not the iris is going to shrink or expand, we also know things like interest or the presence of specific chemicals in our system can impact how responsive the iris is. If you ever look into the eyes of somebody staring at something very beautiful or very intriguing, you might notice that their pupils are significantly larger than you might expect just based on how bright it is around us. And that's because we do actually have a neural feedback loop that allows the iris to shrink in situations where we find ourselves very engaged, even if we might think that we could be potentially overstimulating the eyes for a short period of time. You might see this in cats or dogs when you get them very excited because they have the same sort of override response when they get really worked up or kind of, I guess, attentive to a specific situation. Another thing that doesn't necessarily cause the pupil to permanently stay wide open, but it does slow the responsiveness of the iris, is if we ingest specific chemicals. Depressants are one of the big ones that we know can really slow the functioning of the iris. In fact, this is why you might have, if you've ever been pulled over in the middle of the night while driving, had somebody flash that bright flashlight in your eyes to check how much alcohol or whatever drug you've consumed you've had over the night. And that's because we know when we flash light in the eyes of individuals who've had large amounts of alcohol or some other substance, that their iris will have sort of a delayed response. It won't be able to react to that light the same way it would if we didn't have those chemicals in our system. And these are just, of course, two examples of things that could potentially override the iris. And they're also really great examples of the interplay between the biological structures of our eye and other components of the mind that maybe we wouldn't naturally link to something like the eye. And this is true for a lot of other structures that we're going to cover later on. Another one that sort of has this nuance to it, but we have to take a long journey to get there, is a structure called the cornea, which is at the front of the eyes. And the cornea's primary goal is to just kind of protect our eyes from all the things in the environment that are out there. If we look at the inner structure of an eye, what we see is a bunch of very, not necessarily flimsy, but we'll say sensitive structures. And if damage occurs in a lot of these structures, we can have permanent damage to vision, if not lose our vision entirely. The cornea is a rigid structure that sort of has a nail-like consistency that helps protect a lot of those structures found behind it. But the cornea also does another thing that we're going to focus on a little bit more later on. What the cornea allows us to do is sort of minimize the amount of space in the back of our eyes that's required for the presence of those cells converting electromagnetic energy into a neural signal by bending the light coming from all the different angles and sort of narrowing the scope of what's going through the pupil and eventually what reaches the back of our eyes. It's not necessarily something we would ever notice, but it does have a slight effect. If you've ever looked through a telescope, you might notice that everything seems a little bit bent and stretched in different ways. And that's a byproduct of the lenses that you use for these telescopes or periscopes or whatever you're looking through that uses a lens that kind of creates this sort of skewed image. Well, the cornea is doing that, but we never really notice that it's doing it. Why that's the case is something we're going to talk about in just a few seconds. But before we get to kind of the way our brain can sort of overcome this weird alteration of these electromagnetic signals, I want to focus on another structure that's hidden just behind the pupil. It's a sort of glass-like structure in terms of what it looks like, even though it's certainly not the rigidity of glass. That's called the lens. The lens is very aptly named because just like lenses that we have for other types of things, what the lens is meant to do is bend the light going through our eyes so it can hit the back of our eyes in a very specific way. Now, again, since we're not in an in-person class, we don't get to do some of the fun activities that I like to do here. But if we were in an in-person class, what I would have individuals do is stick their thumb out at me in the front of the class, but have their thumb just a little bit below my head. And I ask the class to focus their attention with just one eye open on their thumb and recognize what I look what I look like as they're focusing on their thumb. Now, most of the class will usually indicate that I'm a little blurry as they're looking at their thumb, but they can still see things like the color of me and the general shape of me. Then I ask students to focus on me with their thumb still extended. And what they'll find very quickly is that if they sort of turn their attention a little bit to their thumb while still focusing on me, their thumb is blurry. Structure's there, color's there, but things are sort of out of focus. Well, these changes in focus are a byproduct of movements of the lens. And as we survey an entire room and look at things of different distances, the reason why all of these things tend to be fairly clear despite how far or close they are is because of the way our lens is constantly adjusting to all the different distances of things that we're trying to look at. I know some of you might be sitting here as I'm talking about this, complaining that you never see things super clearly. Well, there's a possibility that it could be a malfunctioning lens, but there's a better chance that it's probably a byproduct of things like the structure of your eyes or other things that could essentially happen as a result of some type of damage or kind of just a biological reason for kind of the focus of light not working correctly in your eyes. Sometimes there's corrective surgeries that can do things like shaving the cornea or adjusting other things, but for the most part, the vast majority of the population that at least can see, the lens is doing its best almost all the time to make everything we're looking at as clear as possible. And that kind of constant activity of the lens is something we overlook, but it's something we really want to appreciate. Another thing that we can really kind of appreciate about vision when we look at the lens is the fact that for the lens to do this, the lens to bend the light, what actually has to take place is a really weird conversion of that energy that passes through it. We've known long before we were able to actually dissect the eye and look at the lens that what should potentially happen when light passes through the eye and gets to the back of the eye is the image hitting the back of our eyes, because of how lenses are structured and how light bends, we should see essentially an inverted image hitting the back of our eyes. This led researchers to ask the very obvious question of, well, if the lens is inverting the images of the electromagnetic energy in our environment, how is it that we don't see the world around us as inverted? We could also link this to the cornea that we talked about before. If the light that's coming in through our eyes is bent and manipulated, how is it that we don't see it in this weird, bent and manipulated way? And if we want to even add more wood to the fire, we can identify that we oftentimes have lots of imperfections in our eyes and lots of things that float within the different structures of our eyes that we haven't even talked about yet that could potentially make what it is we're seeing a very imperfect thing. So how do we see things clearly? How does that inverted image get flipped? Well, before we had a lot of tools to actually look at specific areas of the brain that might be converting this image, what many scientists did was just come up with tools that could replicate this activity. One of my favorites was a pair of glasses that researchers would put on individuals that would actually replicate the inverted image that was happening as a byproduct of the lens, but it would invert the image before this information was hitting the eyes. This created some really fun games for researchers to kind of have people experience what it would be like to have the world essentially flipped upside down on them if their lens wasn't working. But it also gave researchers a chance to explore how quickly people could adjust to these inverted images. And we're pretty amazed to find that people who would try playing a game of pin the tail on the donkey or playing a game of catch with these glasses could quickly start to adjust to this new world that they were seeing through these special pairs of glasses. They'd often find within minutes people could usually start to acclimate to things, and within a couple hours people would start to see, oddly enough, the world is right side up even though they were still wearing these glasses. And eventually they'd get to a point where they couldn't even recognize that things had been distorted as a byproduct of the glasses that they were putting on. And of course, these researchers, just for fun, would take the glasses off of individuals and find it fairly amusing when these individuals would then report, even without having glasses on them, that the world they were looking at somehow seemed inverted. Now, rest assured, this would not be a very long-term thing. It'd be very temporary. Within minutes, most people would see things fairly upright and not inverted anymore. But it gave us sort of an insight as to how the mind was constantly adjusting for these abnormalities in what it was we were seeing in the environment. And as we got better with a lot of the ways that we could look at the brain through things like fMRIs and EEGs, we were able to narrow down specific areas of the brain that were responsible for this course correction. And what it was able to give us an understanding of is the fact that a message reaching the back of our brains is already, by the time we start to process it in the very introductory level, being manipulated, being altered to an extent. So what we see, at least in our minds, what we perceive, I guess we should say, is sort of a recalculated view of the messages coming from the eyes. It's a byproduct of all the calibrations that we've had to make to accommodate for the lens and the cornea and all the individual imperfections we have in our eyes and other things that we tend to overlook, like the presence of our nose or eyelashes that we don't really want to see, so our brain just finds a way to sort of delete it from the images that our mind is processing. And all this, again, highlights how even though we tend to think of the eye as just this isolated thing that's designed to send information back to our brains, there's actually an interplay between the mind and the eye that's going on all the time that we need to appreciate to truly understand how something like vision works. And this leads me to the last couple stops along the way before information reaches the back of the eyes. If we talk about the structures of the eye, we've pretty much covered all the big things that you need to know, the things that convert that electromagnetic energy into a slightly more complex signal that reaches the back of the eyes. We also have some structures that are worth mentioning because they really allow the eye to maintain its shape and functionality. There's this liquid that's found between the cornea and the iris that's called the aqueous humor. Something that's worth mentioning because it helps keep the front of the eye functioning and moist and keep it, well, in a healthy condition so we can continue using the parts that we use. We also have this liquid called vitreous humor. That's sort of the consistency of something like Jello meets pudding that's hidden behind the iris and keeps our eye's shape and keeps it inflated. And this vitreous humor, which makes up what's called the vitreous body, is essential for our cone, well, I almost gave it away, the cells in the back of our eyes to be able to do what they do, and the space between things like the lens and the cornea in the back of our eyes stays consistent. What those things are in terms of the kind of critical junctures of the eye is really just gateways, paths that the electromagnetic energy has to travel before it reaches the back of our eyes in an area that we call the retina. The retina is a term we use for the back of the eye, the area in the back of the eye, where we have cells that can convert that electromagnetic energy into some type of neural signal. There's also a specific area along the retina that we tend to focus on a lot called the fovea. You also see it sometimes listed as the fovea centralis. This is the area in the direct back of our eyes, where if we focus our attention on something, look at something directly with one eye, we're kind of directing the thing we're focusing on to the light from that thing to this area. The fovea centralis is a really critical player in vision. And to understand that, we have to understand finally what the cells in the back of these eyes are doing. So what are these cells in the back of the eye? Well, one of the cells that's found throughout the back of our retina, including that structure that we call the fovea, or the area we call the fovea, are cells called rods. Now, they're very adeptly named rods because if you look at them under a microscope, they actually look like tiny little rods reaching out from the back of our eyes. These rods seem to be specifically designed to pick up on just subtle changes in electromagnetic energy. They're exceptionally responsive to these changes in electromagnetic energy in dim light. And if we look at how they function and the signals that they send from them, you seem to have a pretty clear indication that the rods are the cells that are helping us pick up on small changes in movement, the presence of things in not only dim lights, but really all types of light. They're not necessarily helpful in picking up on the little nuances of what we're looking at, and they're not very adept at picking up on something like color, but they do seem to be very helpful in just understanding that the presence of things in our environment. And these rods are present in not only human eyes, but many other species around us as well, and they seem to be kind of an essential thing for the utility of vision in terms of survival. If we're looking at the specialized cells that really help humans see all the amazing things that we usually have the ability to see, it's another set of cells that are found primarily in the fovea called cones. And much like rods, they're aptly named cones because they happen to look like cones underneath the microscope. Now, these cones are essential, we think, for color vision. And we're going to talk in our next class about how they actually are capable of helping us with color vision, but just understand for now, they're critical for color vision. They're also critical for fine detailed processing, so that they can really pick up on the fine lines of things that we're looking at. And again, they're located primarily in the fovea. They're centered there, even though there's a few sparse ones found throughout the retina. They're mostly in the fovea. And this is why we often find that color vision and our ability to pick up on fine details is usually dedicated to that area in the direct back of our eyes, where we tend to use it for focusing on things in our environment. And throughout most of this retina and the fovea, again, we have these rods and cones. There is one small exception to this, though. There is a small area along what we would consider the retina that doesn't seem to have any rods and cones. And this creates something that biologists call the blind spot. And it might seem like a problem, but in actuality, it's sort of a necessity for vision to work. It's the area where the messages coming from rods and cones, and actually some other cells that we're going to talk about in our next class, come together and get sent out of the eyes on their path eventually that will then get to the brain, or the back of our brains, in that area that we talked about in our previous class called the occipital lobe, or sometimes again you'll hear it called the visual cortex. Well, it's important to note that when we talk about the path from these, again, optic nerves, or what again we sometimes call in the eyes blind spots, to the visual cortex, that there's a lot of stops along the way before that information reaches the back of our brain. As information leaves our retina, travels down these optic nerves, eventually meets for both of our eyes in a structure called the optic chiasm. This area is sort of a channel for the visual information that helps us partition out the visual information coming from both of our eyes in a very important way. To understand this partitioning, we have to go back to something we discussed in our previous class. You remember back to when we discussed the functionality of the parietal lobe and the frontal lobe. We talked about a process called contralateral communication, this weird phenomenon where the left side of our body is usually processed by the right side of our brain and controlled by the right side of our brain, and the right side of our body is controlled and processed by the right side of our brain. I think I said that right, sorry. Essentially, it's the opposite sides are linked to each other. Well, this is true for touch and movement, and it's also true for vision, but in a slightly more complex way. So it's not that our left eye is entirely controlled by the right side of our brain, and our right eye is entirely controlled by the left side of our brain. Instead, when we talk about vision, we talk about this thing called visual fields. To understand visual fields, you have to imagine looking straight ahead and having an imaginary line shooting out from our nose straight out, so that the world we're looking at is split into two halves. Everything on the left side of what we're looking at straight ahead, and everything on the right side. Now, mind you, if you look to the left, you're switching your visual fields, you're altering it. So that the left visual field skews over to the left more, and the right visual field suddenly comes over to the left as well. Visual fields are about what's on the left side of whatever it is when we're looking straight forward, and the right side of whatever it is when we're looking straight forward. Sorry, it's probably easier to understand this if you actually saw me making the hand gestures that I'm doing right now, but some would be it. Hopefully you can understand this. So when we talk about this contralateral communication for vision and visual fields, what that means is that instead of, again, our left eye being processed by the right side of our brains, it's our left visual field that gets processed by the right side of our brains. If you want to see that represented in the image that you see in front of you, you'll notice that the left and right eye have pink lines on the right side of the eyes. That's because information that's hitting those sides of the eyes, because of how images pass through the eyes, is actually coming from the left visual field. So both your right eye and left eye are processing information from the left visual field. The same thing is true for our right visual field. You see with the black lines that the left side of both our left and right eye is processing information that's in black, and that's the information coming from our right visual field. When that information meets in this optic chiasm, it's filtered so that the information leaving it is no longer about what's coming from the left eye and the right eye, and it's now about what's coming from the left visual field and right visual field. So you'll notice that all the information in the right visual field that again was hitting the left side of both our left and right eye travels to the left side of our brain. The information from our left visual field, which again is hitting the right side of both our left and right eye, is eventually processed in the right side of our brains. And this is all thanks to the amazing filtration system that happens at the optic chiasm. After information passes through the optic chiasm, it has one more really critical spot that it has to go through before it gets to the back of our brains. It's a structure in our brains, a subcortical structure in our brains called the thalamus. And the thalamus is actually a very important system, or I guess structure, for all of the different sensory systems we have, minus smell, which for some inexplicable reason doesn't pass through the thalamus like all of our other systems. Everything else when it comes to our sensory systems, both our sense of taste, taste does go through it, our sense of touch, our sense of hearing, and our sense of vision all goes through the thalamus. And what most researchers have contended is that this happens because this is sort of our gateway to the brain that helps us filter stuff out. We think about all the stimuli that are present in our environment at any given time. We have to recognize that we are being bombarded at any given moment by a lot of stimuli. We would be overwhelmed if our brain tried to process every single stimulus that we were converting into energy at any given point in time. So to prevent us from being overwhelmed and having to try to process too much, the thalamus sort of acts as a gatekeeper. It, with sort of a feedback from our mind, thinking about what we need to focus on and what we need to ignore, allows certain things to come in and prevents certain things from reaching our brain where we would have to process it further. This thalamus also, which is kind of cool, and you might learn about this more in a cognitive psych class, can sort of be overridden by specific stimuli. So you might notice that you may be not processing something in your left visual field because you don't think of anything important there. And if something comes flying at you from that left visual field and looks like it's heading towards your face, even before you realize it, you might be dodging that thing moving for you. In fact, sometimes we can sort of embarrass ourselves with some of the overrides that happen. You might have somebody who gently tossed a piece of paper or a feather at you, and you acted like it was a baseball coming straight towards your face that might kill you if you didn't immediately duck out of the way or jump. That's because this thalamus isn't necessarily designed to really process exactly what it is that's coming at us. It's just designed to kind of not only filter stuff out, but kind of fast track things to other parts of our body so we can react to the dangerous stuff, the critical stuff, before we even really know what it is we're reacting to. It's a really cool structure within our brain that merits a lot more discussion than we're able to do in this class. And I hope you learn more about it in future classes. But for our purposes here, just understand that it's something that really does limit the amount of visual information that eventually makes it to the back of our brain. And the thing that's limiting it is things like our attention to certain stuff and our decisions as to what's important, what's not in a given situation. But the information that does get through the thalamus and reach the back of our brains then typically goes to the very back of what we call the visual cortex. And this is where the perceptual part of sensation and perception really starts. If we get into a future neuroscience class or a biological psychology class, what you'd learn about, and we're talking about perception, is all of these different structures found in the brain that are responsible for specific types of visual processing. One of my favorite things to do in my sensation and perception class is to really break down all the different steps along the way so we can appreciate every nuance to what our brain is doing at any given point in time to let us understand the world, the visual world around us. You'd learn about V0, V1, all the way up to V7. You talk about the what and the where path. You talk about these really cool structures that are responsible for things like which eye saw what and what color things were so we could better understand things like depth and size. But we again are in an intro class. I'm looking at the time and realizing we are sort of nearing the finish line here for this specific lecture, especially because I don't want to go over the 50 minutes that we've been doing the last two lectures. So understand, that's for future time, but again, when we get to that stuff, when we get to the breaking down of that information, we're really talking about the process not of sensation anymore, but of perception. The electromagnetic energies that have hit our eyes have been converted into a neural signal, and now we're finding a way, because it's reached the back of our brain, to convert that neural signal coming from our eyes into some type of information that we could actually interact with. This process of perception, the paths that are taken, and what it is we do with it, is what we're going to be turning our attention to in the next class. It's also important to note that when we move on to this process of perception, we're not just talking about what the brain does anymore. We're talking about what the mind does. And this is why we're going to be moving in our next class away from biological psychology into another area of psychology that's very popular called cognitive psychology. We'll also talk about some other areas of interest and research that kind of came off of this cognitive area when we talk about something called Gestaltist psychology. For now, hopefully you have a better understanding of how vision works and all the amazing structures that come together to allow it to do what it does. And you're well prepared for our next exploration in this topic when we talk about perception and how the mind allows us to really, again, convert those neural signals into something we can use. I hope everything's going well. I hope to see you soon. But in the meantime, take care. Bye.